Faircoin.
Is this on now? Okay. So welcome to the Common Repository panel this afternoon. The topic is the benefits and challenges of sharing AIRR-seq data. And we have 4 individuals who have graciously agreed to participate in the panel. First, we have, let's see, Xiao Liu, who is head of Health Institute at BGI in China. He's also an adjunct professor at Northwest University in China. And he runs a sequencing platform for AIRR-seq data and also operates the Pan-Immune Repertoire database, which has almost 10 billion AIRR-seq sequences in it. So that's a marvelous resource for the community. Then we also have Anton Langerak, who is Associate Professor of Immunology and Laboratory Specialist in Medical Immunology at Erasmus University Medical Center in the Netherlands. And he's a coordinator of the Euroclonality NGS Working Group for the Euroclonality Consortium. And their primary aim is to design, standardize, and validate both assays and analysis approaches for AIRR-seq data. And then we have Bryan Briney, who is Assistant Professor of Immunology and Microbiology at Scripps Research Institute and is generating some really fantastic, huge AIRR-seq data sets. And then we have finally Konrad Krawczyk, who is Chief Scientific Officer at Natural Antibody in Germany. And their primary aim is to work with pharmaceutical companies to develop safer antibody-based therapeutics. But he was also a developer of the Observed Antibody Space data resource, which currently has about 500 million IG sequences from about 55 different studies. So also an excellent sequence resource for the community. So with that, we'll just turn it over to the panelists. Would you like to say something, Christian?
Yeah, so first of all, of course, thanks for joining us here. And so probably in analogy to what we did yesterday, so if each of you could briefly comment on your personal view on the benefits, but also the challenges of sharing AIRR-seq data to the broader community.
Okay, thank you. So, yeah, I may get started. So, so my point of view, for sharing AIRR-seq data, some two important issues should be solved before we are sharing any AIRR-seq data. We talk a lot about these issues. Firstly, we have to think about what portals, what repositories can we use to share this data, right? We have a lot of different, repositories. We talk a lot this morning, AIRR-seq and others, and some of them have a huge data, but they're quite scattered around in different databases. So which one should we use to have a centralized database, or can we, use API to collect all of them? That's the first issue we have to solve. And the other thing is that after we share our data, so how can we use those data? So we need formats, we need the minimum standards. We talked about this morning, right? And what can we use to, to meta-analysis to reuse this data for the deeper mining of this data set? So that's something we have already discussed and we're working on it, but we haven't solved them. So that's an open question. But I think that before we can, make full use of the interchangeable data sets, we have to solve the issues. At least we have the solution for them at the first end.
Yes, I'm participating for the first time in this community, and I'm quite impressed by all the things I've heard. So in terms of data repositories and minimal standards, I think there's already a lot that has happened, which is good, because talking as a academic, but also representing, as was already introduced, a network of people working here in Europe on clinical applications. I think it's very important that we clearly define between each other, and that's what we already experienced in our network, but I think it holds for the whole community, what the important issues are. Talking about benefits of sharing data, I think it's obvious from a scientific standpoint that it's almost a duty to share data. However, because there's various applications that you can think of and data analysis that you can think of, using validation sets, reference sets of data, also for not only for research but also for clinical applications. However, having said that, I think it's key that we have fully annotated metadata, and I think we've heard a lot about it already in the morning, which is good. And I think that's also still one of the challenges, because if the data are not fully annotated, I think that's a shame. We can't do too much about it. And other challenges I see in the context of the applications we are working with is more at the side of GDPR issues, already alluded to this morning as well. I think we can go a long way and solve a few things there, but still it requires our attention. And the other thing is that maybe a special thing in the context of the field I'm working in is that we generate a lot of AIRR-seq data in the context of clinical trials. And what we see is that some of these data are sometimes not open for the public, because pharma companies are interested in those data and kind of protect those sometimes. So I think there I see a challenge for our field also to try and go around it and make that more usable for other people as well.
Yeah, so I obviously agree with both of you guys. So I think we all know about the issues with technical issues with sharing things, right? So repositories, where do they go? And formats should all be complementary, and tools should all be able to handle the same formats and all of that. But one thing that I think gets lost in meetings like this is that there's still a lot of people who don't see the value in sharing their data, right? So I think a lot of those people aren't at meetings like this about how to share data, right? So I think it's important that we also be advocates for what the value proposition is for sharing the data sets and having these huge data sets available to everybody. I don't think it's, I mean, preaching to the choir here, right? You guys are all interested in that kind of a thing, but I think there's a lot of people that are not. So I think it's important to remember that.
Hello, everyone. Thank you for having me on this panel. So as introduced, I'm sitting here because I was a senior author on the observed antibody space, for which the heavy lifting actually was done by Alex Krawczyk here. Raise your hand. Thank you. So basically, I fully agree with everyone else that generally, like, data needs to be properly annotated, cleaned, because otherwise, you cannot do anything with it. And as right now, like, there is a drive to do proper machine learning on it. If you have erroneous data, then you're basically just going to learn a lot of errors. And I can just tell you from my personal experience from just working with pharma companies, there are many companies that actually just reach out to us to grab a lot of publicly available data. They have a lot of data of their own, but they do not want to share it, for obvious reasons, Like if you have your own phase-specific library, then you do not want to your competition to actually see what molecules you're going to be making out of it. Having said that, of course, there is a lot of dead data in companies. So everything that is just past patenting, past commercialization, and still the companies just just to be sure and they do not want to release it. So generally if there is anything that I think that is coming out from meetings like that is just raising awareness that we really need to clean data properly, and this is why all the working groups, common repositories, the efforts such as AIRR-seq are actually really good and actually raising everyone awareness that they should actually make the data usable, because otherwise there is no point of publishing it.
So I guess now we will open it up to general discussion and questions from the audience. General discussion, questions from the audience, but not just questions, also your own thoughts and comments on this topic. And I guess we have someone running around with a mic. Okay, great.
Well, so since we have everybody here, maybe could we? I'd like to hear a little bit, like, how does your euroclonality work, and what is, what are your purposes in, just as a general idea?
Well, thanks. I'm happy to explain. So we started a couple of years ago, like 5-6 years, when we realized that in the next era of clinical applications for antigen receptor analysis, the NGS technology, of course, had to be implemented. And at that time, there were proof of principle studies showing that it would work. And so our aim was not so much to try and prove that, but rather to work as we did in the past in the old day assays, I would say, on standardized and validated approaches, because that's something that unites us as a group. We are interested in the applications and all the special issues around that. And so we started to work on developing assays for the whole spectrum of antigen receptor analysis, which includes the complete rearrangements, which have been discussed a lot here, but also incomplete rearrangements in the heavy chain locus, but also including kappa special rearrangements. For those familiar with that, in kappa there's inactivating rearrangements with the kappa deleting element. And those are not so commonly analyzed, so we had to develop not only the assays, but also further algorithms for analyzing those. So along with the wet lab work, we had a bioinformatic pipeline development, which is AIRRC interrogate, and that is intimately interconnected with the wet lab analysis. So what we've come to now is a point where our first studies have been accepted, will soon appear. They are now in press with leukemia, so that we can share that also with the broader community.
It's more of a consortium, or it is an open database that, I mean, what exactly is the level of?
It is a consortium. So in terms of the data itself, they are open, but the data we have so far is mainly around pilot studies, and of course in the context of generating data with these tools within our consortium or outside, new data sets will become available. But then this comes back to a point that I made before. Sometimes these data are being generated in the context of clinical studies, so they're not so open as maybe we would like them to see. But yes, so we are generating data, and we're getting to that stage now.
So it's a mix of open and other types. So.
Okay, so while everyone here is still brooding for the next question, Felix, there is a...
Good afternoon. Thank you for having such an interesting panel discussion. I just want to share my thought on the data. So the data is a strategic advantage of a laboratory in contrast to other laboratories. And I think this is the most, pain point in sharing the data. Why do you want to share your strategic advantage in terms of publication, interesting software tools, which leads to more grants and more funding? What do you think about this? What do you think about vision that the data is a strategic advantage? And that's why people don't want to share it?
for that. Yeah, I think you're correct that that is a fairly common mindset, right? So the data you generate is a means for you to get more money to generate more data and papers and that kind of thing. So that's correct, obviously. So if you have data that no one else has, you can do things with it that no one else can. So I think one thing that's important is not to discount that as a concept, but to make sure that people are aware of the advantages you get from sharing, which are different than the advantages you get from not sharing, but at least I think more important, right? So one simple example is my lab does a lot of sequencing, and we also do a lot of tool development. So one thing that we really would like to have is like well-validated kind of ground truth data sets, right? So if we can contribute all the sequencing data we generate kind of from normal sequences from people, and that helps build these validation data sets, that's worth it for us. That trade-off is worth it. So there's a lot of things you can get by sharing and having these huge common data sets you can't get otherwise. So I think making the value of that more clear to people would help. So you're not gonna be able to convince them that it's not worth keeping private, right? There's always, those advantages are clear, but the advantages for sharing tend to be not as clear, I think.
Okay, thank you. And what is your idea? Sorry for interrupting. What is your strategy to change the mindset of researchers? Because in this case, you need to change from the mindset from valuing the data to valuing the results from shared data, and that's quite a big change.
Yeah. So I think. If you look at it kind of from a software perspective, I think if you look at open source software, for example, right? I think the thing that can convince people to get involved in that most is seeing an ecosystem that they're benefiting from, right? So if I'm writing software and I'm using a bunch of open source tools, I get the feeling kind of innately that I should be giving back also, right? So I think the best way to start it is if the groups like this that are already kind of on that side contribute their data and build this kind of infrastructure. Once people start using it, they'll start feeling the need to kind of contribute back, I think. That's at least hopefully how it will work.
I agree, and I think what we should do also is show the examples where there's really a clear benefit from sharing data. And I'm thinking about situations where bringing the work of different groups together on small numbers of a certain disease entity or a certain condition can only result in meaningful data if you do that collectively rather than each and every one on their own. And I think there's a good example from a network I'm also involved in, and that is the European Research Initiative on CLL, where a group on IGCLL, as it's called, is working together, collecting CLL cases from all over the world, now mounting up to 20,000–30,000 of cases, where only then you start to see certain subgroups popping up, which you otherwise wouldn't see because you would literally have one or maybe two cases in your own cohort. So I think it is important to show that such examples are around and that they show the benefit of working together and sharing data, which is completely in parallel to what you're saying, that there is also a need to have your private benefits from your data, because that's really the system we're working in.
Maybe just add one thing to that. So of course, like, I think that there is a difference between a strategic advantage whether you're in academic setup or whether you're in industrial setup. Yeah, whereas like in industrial setup, like, it's very clear, yeah, that like if you have data, then you should really just not release it. Yeah, because like, why should you do it? In academia, I think like, it's pretty obvious, yeah, because if you make your data usable, then you make your work more citable. Hence, like, you're gonna get more grants. Yeah, so basically, like, a good example, like just Bryan's work, like, because he made his data available and easy to use, then like people are already citing us included. Yeah, so if you basically can make your data available so people can trust it and reuse it, then it's a win-win situation in academia for you.
Yeah, that's actually a great point. Sorry about that. It's actually a great point, especially for like the tool people, software tool people. I mean, one thing that's much, much better for you personally than having a tool that no one else can use is making a tool that everybody uses, right? Like that's something you can get grants with, more than having this tool that no one uses except you, right? So yeah, I think that data can be the same way, right? If you're generating amazing data sets that people are using, you can get a lot of funding to keep doing that, right? So yeah.
Yeah, I totally agree that just we mentioned about the things should be divided into the academia, research, or the industry. So we all know that for academia research, we have to share data after publication. It's required by most of journals, but currently, in current situation, that just like other data sets, not just L data sets, that people, data owner or data generator are not too willing to share the data because we don't have protocols to encourage that activity. So what people will gain from the sharing data after they get publication, they get cited, they get something else. So we have to think about how to encourage them to share the data. For example, that if the other people use the data to do some of the, applications or useful discoveries, not just citing the data set, but also, to try to, to encourage from other ways. So we should think about that. For the industry point of view, that's more complicated. For the they devote resources, they devote samples. So what they can get from the sharing data? That's an open question.
So, Encarnita, do you have a question regarding to this one, or do you want to go to another topic?
Well, it was kind of related, but maybe going to another topic. The idea was, so I think I totally agree that sharing data, it's important and we get a lot of benefits. However, so Anton mentioned the issues with clinical trials and with patient data. So AIRR-seq data are sequences, so genomic sequences. So how can we rule out the limitation that we have maybe with the GDPR and some hospital, or at least I know that from APHP in Paris, we are not allowed to distribute publicly patient information, patient data. So do you have anything in mind with this? How could we do to try to make things easier?
I have a question, a follow-up question. Under the GDPR rules, is the sharing strictly forbidden, or is it permitted if if it's included in the patient consent process for participation in the study?
It's allowed if you, so Article 9, which is the main protection for sensitive data, allows you to share under point 1 if there is sufficient informed consent. So if you get people to sign that form, you're good to share. The question is a little bit on how far you can go, because some of the usage purposes you might be you might want to have in there might not be specific enough. So I know that, for example, in Germany, there's right now an initiative that is evaluating this concept of broad consent, which is a little bit difficult there, but they now seem to come to a conclusion that this seems to work, and so this of course then at least would apply to all of Europe, and then probably we can I think people try to evaluate similar instruments also for North America.
Maybe before you go on to the next topic, I had never heard the term dead data, but I think that idea that companies may have a whole bunch of data that they're just holding and they don't really know why, and if that might be a way to get into sharing data from industrial partners. But I also question this distinction between academic and industrial partners. The recent academic colleagues that we've talked to, they're all, they have this, they've got this, they're in between, and they have certain data they can share and certain data they can't share because of industrial partners. It gets complicated and confused very quickly, I think.
So I just had a follow-up question on the consent is it typical then for sharing to be included in informed consent processes for trials in Europe, or is that unusual? I mean, we were talking about what kinds of things can we do as a community to facilitate sharing, and one thing that had been on the Common Repository Working Group's agenda since one of the earlier meetings was to develop informed consent language that would facilitate sharing under different jurisdictions, and that's something that we have not done, but I wonder if that's something that is worth resurrecting as an effort.
So before, this is Mike Sterlington from 10x. Before I came back to repertoires at 10x, I was heavily involved in the Human Cell Atlas, which is all about data sharing, and we spent a lot of time thinking about informed consent and getting to the point where we could put language together and consents together so that we were able to share full RNA sequencing and genomic data from our donors. It's certainly possible. What we found was that people were often limiting their consents because they were just not putting the right language into the forms and were putting the usage scope, limiting the usage scope too much. And we worked to, sort of with our donors from the organ transplant network in the UK, to really broaden that language out so that it was informed and suitable for all of our purposes, and we found that that was very successful. So I think it just requires that communication and foresight to make sure that the language on the consent forms is suitable for what you'd want to do. That's a very UK-centric perspective. I don't know so much about the rest of the world, but I know the community in the US, for example, are working towards similar goals to really make it so that it can be openly shared.
Great, thank you. You have someone back there, Felix. Felix. There's
So I guess a commonality between you to some extent is you have very large repositories of data. And so one small question is, is there any overlap between them? Or, have you guys consumed data from multiple sources and put them together, or is it all unique data sets? But the more important question that I, would want someone to answer is, could you get me excited about large repositories of this data? Like, what can you do that is so special or awesome now that you have like 10 billion antibody sequences, other than get extremely small error bars on, like, the V gene usage histogram or something like that?
Somebody's very eager.
No, I am not excited, because the more data you have, the more noise they are. So, and at some point you have so much noise that you can't distinguish a signal from it. So on the more serious note, I think before talking about the very large scale data, we definitely need to talk about how we pre-process it and remove errors from it.
But I'll repeat my question. What is exciting about getting a gigantic repository? I know some of you have billions of sequences.
Well, maybe I can answer, at least from my perspective. I wouldn't get excited so much about big data sets per se, but only if they are very well annotated, very specifically annotated for a specific purpose. And I think that's what I try to indicate with my example of bringing big data together and then sorting out very rare types of subgroups or conditions that you're interested in. But I think key for me to get excited about big data would be that they are very well annotated and completely annotated, because otherwise they're basically meaningless. Sorry? What is missing? I think that's different. Sometimes what's missing is maybe the type of cell something was taken from, whether that's a naive or a memory cell. Sometimes it's maybe the age of the individual, which influences a lot what you're seeing. So I think it can be quite different. I'm not saying that a lot is missing. I'm only saying that anything that is useful should be quite complete for the annotation you are interested in.
Okay, so regarding your first question about the overlap of the different repositories, so we are, I don't know for other guys, but I can talk by myself, about our groups. So we have a a database that's mostly the data generated by us, and we have a initiative, a consortium. We're working with the hospitals, with the research institutes, and some of the industry partners, that we get samples from the hospitals and get full comprehensively annotated clinical information from them. And we got some funding support from the research institutes. So we generate data, and we are currently in our pain email repertoire database, we have the almost 2,000 samples that's already shared with the public. And we are also working with the AIRR-seq. I think in the future we are going to work together to how to share the data together. So from our database, we are mostly generated data, and we are put our data into our databases. But we also have the plan to work with our receptor to put the other data together. So the point is that we can use different repositories easily to get all the data together. So that's
So if I could, an example that I think about a lot, within the last year, there was two studies looking at melanoma and anti-checkpoint therapy, and they used the same drug, and they were looking at adverse reactions, and they were trying to predict, what patients would have adverse reactions. And the two studies came, and they were looking at expanded T-cell receptor repertoires, and they came at the exact opposite conclusions as to what the markers were going to tell you about who would have an adverse reaction and who wouldn't. In my mind, that's because we need to put 10 of those studies together and see who's really right. I mean, or maybe there's no signal at all. But we need to put, larger sets together to look at reproducibility to ask, was that contradictory result because that's the true state of the affair, or is it just they didn't have enough? We need 8 more studies to give you. So that's my goal.
Yeah. Well, so I think one thing is, a parallel for that is like genomes, right? So why is the 1,000 Genomes Project better than the 10 Genomes Project, right? And I think the important thing as you start to scale bigger and bigger is that cohort selection becomes a big priority, right? So sequencing 1,000 random immune repertoires is not super informative once you've sequenced a few, but targeting groups in terms of disease states or ages or whatever the thing is you want to do, I think that's one way where a common repository can help, is because you have people doing all of these smaller scale experiments that get put together, and now you have these huge diverse cohorts kind of all in one spot, which that's how I view it as being beneficial, but we'll see if that actually ends up being that way.
Okay, so if I remember the questions, the 2 questions. So first, if it's are there any overlaps between us? And second, to get you excited, why we're actually, well, what can we get out of it? So yeah, of course, if there are overlaps in public data. So this is why, for instance, we have in observed antibody space, we have many studies that are also in AIRR-seq. Yeah, so this is why it's very important, in an effort like this meeting, to actually get everyone to just share and coordinate efforts. Second one, to get you excited about what we can actually do with it, it's precisely building on Bryan's point. Yeah, why 1,000 genomes is better than, I don't know, 10 genomes. Yeah, so if we know that the space allowed to antibody sequences is huge. Yeah, but if we knew that it was uniformly, if it were a uniform distribution, yeah, then just sampling more and more and more and more and more wouldn't be that interesting. Yeah, but we know that basically there are some strategic preferences. Yeah, the immune system doesn't really just produce a totally uniform distribution. It's actually there are strategic islands everywhere. And basically, like, as you get more data, then you can actually just, identify like what are the strategic preferences in developing and defining the diversity of your antibodies. Well, I'm speaking from an antibody perspective. I realize that like there are also many TCR people here. And of course, like, also, so this is from the academic perspective. From industrial perspective, of course, like, you want to know if you have more data, then you know how antibodies, like what is the natural diversity allowed to antibodies. If you know what is the natural diversity allowed to antibodies, then you can counteract many many bad properties of artificial sequences. Yeah, you know that, like, for instance, in this position here, you should never have a glycine, yeah, because it might be immunogenic or something. So this is why actually, like, getting more and more and more data is informing us better. But of course, like, building on the previous point as well, more data might also mean more errors. Yeah, so this is why it's very important to actually like have it clean.
I just wanted to emphasize something that was just said by Konrad and Bryan, which is that I think the data really become useful at that scale when the annotations are rich about the individuals. So it's definitely important that we know, what cell type was used and, all the details of the study protocol, but I think we need to have the richest possible annotation about the human subjects in order to really start understanding how particular phenotypic traits or clinical traits are associated with important patterns within the repertoires.
Okay. William?
So suppose that I have data and you've persuaded me that I really should contribute it. Where would you advise me to deposit annotated sequences today? And if there isn't an answer to that, what should we do about it?
There are many answers.
Yeah, there's a lot of answers, which I think means there's no answer, right? So, I mean, right now, the way everyone's doing it, it's to kind of roll their own thing, right? So we have like links to the process data in a GitHub repo, right? Or you can deposit all the raw data in the SRA, but that's not super helpful for someone who wants to do all of the processing steps that you did, right? So I think that's where the Common Repository Working Group is kind of coming into play a bit, but you're right, there is no, as far as I know, place to do that now.
So
no single place to do that now.
I think I would disagree with that, though. I mean, I think what we got from the last year of work is we have a place based on at least the NCBI framework where you can put in the raw data into SRA and the process data into GenBank, and they can take the scale of data I think that we've been generating so far. And I mean, I would like to at least see us push on that framework that as a community we've spent quite a while developing. And we'd love to hear sort of feedback on people's experience with this, but I do think we have that framework.
Can GenBank really scale? I mean, so you had mentioned that someone had mentioned that they had like 1.5 million antibody sequences in there that were searchable now. But I mean,
so we
have 10,000 times that in one data set.
We have spoken to them about the scale of data being generated, and they have told us that they are willing to accept that scale of data. And so far, the stuff we've submitted, they've been able to take. So I think we should start. Pushing on them, because I think this is a great supported infrastructure that we can have as a central location.
I mean, so from a repository standpoint, what you'd like to be able to do is search for things like, I want a bunch of sequences from people that have lupus that are between 25 and 40 years old. Is that possible through GenBank? Or is that more of like a sequence search where you can put in something and get something similar?
No, so theoretically these searches should be possible, but I do agree that we might hit a limit at some point, because in the end we still have to be aware it's mainly a genetic database, and the amount of metadata that we can put in, if it's normal metadata, that's straightforward. And I think doing rich annotation to a very large extent is already possible. But once we're moving into this thing where we don't know whether we're dealing with metadata or data, so like if you're talking about single cell, what we talked about today, phenotypes and so on and so forth, then the boundary might be blurry. But this should not disincentivize us now to use this resource to the maximum extent that is possible, because there is I think one important thing that I would like to revisit again in a couple of minutes, and that is sustainability, because this is a resource where we're pretty sure that it will still be funded within for the next 20 years.
So do you think, Stephen, maybe this is like an opinion thing for you, but do you think that if we just pour tons of data in there, that might encourage someone like the NCBI to make something that's kind of domain specific for us? If they're like, wow, we have so much data from you guys that it kind of deserves or kind of warrants its own thing?
So yeah, I mean, so in our discussions with them, for example, you know, they have certain keywords that they support in terms of annotation. For example, like I think CDR3 region was one that we've spoken to them about, where that's not one of their official sort of annotation terms. So we sort of work with them on a way that we could get that in there now, but they've told us if we get enough data in there, they are willing to sort of put those kinds of official keywords into their structure. So they are sort of very open to getting more data from us. I know they're using some of the same resources to get in microbiome data, which is very, very large scale data as well. So I do think that they can, you know, that they will be able to support the level of data that we've been generating.
Microphone.
So well, can NCBI then take linked data from, let's say, a single person? They have microbiome data, they have genomic data, they've got AIRR-seq data. Can that all be linked together for searching on, you know, an individual and multiple individuals?
Yeah, I think in theory that kind of thing is possible. So they have structure where essentially there's multiple different databases. There's sort of bioprojects that describe a sort of a project level information, biosample where you get sort of sample level information. And I think it should be in theory possible to link that biosample data, that biosample record to different kinds of information that are collected.
I think, to be honest, one thing that biosample does not support is a unique global subject identifier, which makes sense from a privacy perspective. So of course you can use the same handle within a larger study, like you did for, so that you reuse that handle, but then everyone would have to use that handle in the subsequent studies. So that's not something that,
it's certainly not built in there.
Yeah, it's not built in there, but that can happen on an organizational level.
Now there are, I mean, that's a challenge. But there are repositories like the import data repository, which is supported by NIH NIAID, the Immunology Institute of NIH. So import itself essentially describes sort of subject level information, and then has links out to other repositories like GEO for when there's transcriptional profiling data, or to SRA for this kind of thing. So that's another resource that's supported at that level that we can take advantage of for linking multiple types of data.
Yeah, Uri again.
Are you still not convinced that these are important?
But coming back to William's point, I think another potential answer is that there doesn't need to be a single place. Maybe GenBank is not the best place, and maybe there is no best place. But in principle, is iReceptor not specifically addressing this exact problem, which is you can dump your data wherever you want, and then there's an API that allows you to search across the places. So that begs the question from all of you of what are you doing to make sure that your data repositories are actually going to be accessible through this single API, because this is in the works, and I believe there is already a draft API, and maybe you haven't implemented it because you don't get first or last authored papers doing it, or maybe you haven't implemented it because you feel that the API is not yet suitable for your purposes, or maybe you haven't implemented it because you just have other things that you're doing. But I mean, what will it take to do that? Or can you commit to implementing it, or are there still deficiencies that we should address for why you have not yet or do not intend to make your repositories accessible in this common way?
Yeah, so we definitely intend to. So it's less a desire issue and more of like a manpower issue for us. So as soon as we're kind of confident that the specification is not going to change and the API is kind of solid, we're ready to do whatever we need to do. So yeah.
Yeah, so basically from our side, I have been in conversation with Bryan from AIRR-seq for quite a long time now. So yeah, basically it is one thing, just a bit of lack of time, but there is a clear incentive to that, yeah, because if all of our repositories are accessible and more usable, then of course then people are going to be citing them more, yeah. So there is this clear academic advantage in that. So basically, going to the point that there are no first and last author publications from that.
So we haven't think about that our database become the most comprehensive repositories for everybody to download what they need. But we just want to construct a database that tend to be unique. That's, for example, that they can link all the clinical annotations. They can use in the same pipelines, and they can provide some functions for the users to download and compare and visualize the data. So we are not intend, to put everything into our database, but for that kind of purpose, that we want to work with the other centralized database to fulfill that.
And we are also still exploring options, so we're not to that stage yet. But I think what we've heard here is a good suggestion and a good way to further pursue.
So sorry, my question wasn't quite as naive as it might have sounded, Yuri. I mean, is it really realistic to think that anyone, any researcher that produces some annotated data is in a position to put up a repository that will last for, even a few years? But your other question I thought was really interesting, and to turn it around, apologies, I didn't realize we got that far with Zhenbang. But it is a good answer. But if it's our answer, how do we link that into AIRR-seq?
So I just want to be a little bit clear here, because we're talking about AIRR-seq, but I think what we're really talking about here is a set of repositories that are implementing the Airdata Commons API. And right now we have an AIRR-seq API that kind of acts in that role that VDJ Server and AIRR-seq use today. But we have an Airdata Commons API that the community is establishing, and that's what we're talking about, right? It's not AIRR-seq per se. So the repositories will implement an API, hopefully, and we provide a repository that implements that API, and we have a turnkey repository that if you wanted to, you could download and it would implement that API. But I just want to be clear, it's not AIRR-seq, and AIRR-seq doesn't have a repository that will take everybody's data. Just be clear about that. And I think it'd be interesting to see if we could link and connect to NCBI through implement an API to query NCBI. And I'm not going to try to answer that question either, but I just wanted to clarify that there's an API that the community is defining, and that's the Airdata Commons API. And so I just wanted to make sure we didn't mix those two things up.
So
there are repositories to which you can't.
Yeah, sorry, actually, yes, good point, Lindsay, thanks. So Lindsay has a point, because Well, so I think, Lindsay, I think if I'm not mistaken, or Scott, maybe you can comment. So VDJ Server is a place where you can upload people. So researchers can go and upload their data to VDJ Server. And through VDJ Server, you can publish data to the public side, which will live in and be queryable by the API.
I think my concern is a little bit the same that William is raising, is that, so if you have a lot of separate kind of individually managed repositories, so what happens if the manager of one of those repositories leaves academia, for example? Those sequences just drop off of AIRR-seq, or you see what I'm saying? So yeah. I think a combination of something like what Steve was saying, where you have
absolutely
kind of a more permanently funded thing, but then building tools on top of that, like AIRR-seq, that can query that in a way that maybe the NCBI doesn't necessarily support directly, right? So I think the combination there might be really powerful.
Yeah, we talked about that in the Common Repository Working Group session yesterday a little bit when we were brainstorming, because, I mean, I don't think we should necessarily think of these AIRR-seq repositories as being long-term curation. I think you always need to go to that standard NCBI. And the way I think of it is the sequences, the raw sequences go to SRA, and that's, you know, that's your long-term curation. And NCBI's kind of this being able to publish the annotated data is a new thing, and if that works and they can support the scale, then great, that's another repository. The way I always think of it is there might be a small number of really, really large repositories. There might be BGI that has a huge one for China. There might be NCBI in the US. There might be EBI in the UK. And they may take this AIRR-seq data, and they may be really big ones, but there might be a bunch of small ones in different labs and things like that. So I think from an API perspective, that's what we want to support. And we want to enable sharing across all of those, with the expectations that there will be some small number of really big ones, some small number of medium-sized ones, maybe some small labs, and we want to try to support that. That's.
And I think the philosophy of stability is, you know, some groups, they try to get a huge central repository, and then NIH has to put millions of dollars in every year to support that central repository. Or you have multiple repositories, and if one goes out, it goes out, but you have another 99 that are there. So it's a different view of, you know, how you get sustainability. And
sorry, we have another couple of minutes, do we? We are until, no, sorry, we're until 13:40. Okay.
I have a quick question relating to the question about why to get excited about putting all these data sets together. And that's, do you think that differences in sequencing strategies and pre-processing methods will pose a problem in terms of finding differences that are specific to sequencing strategy or pre-processing methods and not biological signal?
Yes. So we've actually done, we did like a trial thing where we tried to train a neural network to distinguish between flu antibodies and not flu antibodies, right? And we used two public data sets, one that were flu-specific plasmablasts and one that were just healthy people that were sequenced. And it turns out the model was amazing. It was able to distinguish basically perfectly, and it was doing it by identifying different primer sequences that were used. So it was essentially identifying the region in framework 1 that the two different groups had used to amplify. So yeah, 100%, that can definitely happen.
So any suggestions on how we would know about that stuff? How would these databases incorporate some of that information so that we can ensure that there aren't confounding factors like that?
That's all the metadata stuff, right? So if we have metadata about the strategy used for library preparation and the strategy used for annotation and all of these things, I think that can help. But yeah, I see it would definitely be a problem, I think, if we don't address it.
To directly comment on this, this is, so MyAir does include metadata fields for amplification strategies and also for primer sequences.
Yeah, so maybe if I can just add to this at the end. Yes, basically, of course, like, different ways to get your sequences, like, are going to produce different results. Yeah, like I think that one of the most important ones is actually the error rate, Because if like one technique or like one set of people, they can just produce data that like is pretty erroneous, and the other one maybe they can produce more clean data, yeah. And usually the people that actually like are doing the sequencing, like they have more of an idea of like how good their data is, rather than just us trying to infer post facto like what is the error rate, yeah. And especially like in such mutable sequences like antibodies, it might basically just introduce huge amounts of noise that they're just going to go unnoticed until person that actually like did the sequencing just says actually it's not that good.
So I mean, of course, this experimental or the technological variations impact the results that we have just mentioned a lot. And of course, adding, detailed annotation of these pipelines will be helpful for future, to remove or eliminate these potential confounding factors. But from my point of view, from my experiences, that if you're using the overall, repertoire indices, that will be very much influenced by these factors. But if we use some of the, like states associate T cells or B cells or disease associated T cell B cells, that may be less influence. But that's also relies on that we have the full information recorded.
So I think Common Repository has about 20 more minutes, right? So been great so far.
Okay, so one question that I would like to revisit. So some of the comments that I wrote down now coming from the panelists is, and from the discussion, I think that everyone agrees on. Number one, it's important to have rich and accurate metadata annotation, which is costly to do. Everyone wants to have free data. And as it was already mentioned, so the question is, how do we incentivize these things? And so for Bryan also introduced that concept of the Airdata Commons during his talk previously. And so for those of you who are non-native speakers, if you Google Commons, then typically what you find is the tragedy of Commons, which is this example that a village has some kind of meadow and everyone sends their animals there to graze, and at some point, well, there is no grass anymore, because for each individual player, it's an attractive strategy to overexploit that resource without giving back, because there's a short-term benefit to this. So, and this, of course, is the same thing that that you mentioned initially. So it's always attractive to defect from such a sharing scheme. And I think the question that we need to ask ourselves as a community is, how do we shut down this noise? Is of course, how do we set new incentive structures? I mean, we already discussed citations. That would be the classical thing that we do academically. But if we're looking at, for example, the things that the EU is now setting up with Horizons Europe / FP9, is that they're trying to specifically address this, and they've identified this as one of their 8 pillars for open science. How do we set new structures? They identify it as a problem, they don't have a solution. So I think it would be good for us to see, okay, what are the ways, are there ideas how we can do this besides citation, and besides using Elsevier's metric proprietary score, which just puts another number onto researchers' productivity. So by simply commercializing this whole thing. So what would you think would be potential strategies for this one? And what would be the things where we need to make sure that that the resources really can be maintained long term? So is this personnel, is this infrastructure?
Do you mean how to incentivize it further beyond citations, or?
Well, so there is this clear connection between how to incentivize things, because and how to fund things, because this is how the way how it works. But if we get funders like the EU to the point that they accept things other than just your citation record, then the question would be what would be the format?
Yeah, so I guess like basically like this is maybe just a lot of wishful thinking and just going to the EU and just telling them like how to just recognize these kind of efforts. Yeah, but generally like they're, if from what I still remember like from getting grants, yeah, it is, people look at citations, people look at publications, but they also look at, for instance, like involvement in certain groups. So I guess that like if AIR just becomes like generally just grows and becomes like a very well recognizable group, and if you can basically then use it as a bit of like street cred for getting grants. Yeah, so basically you're saying that like I have submitted that many repositories like within the standard AIR, within the AIR standard, and then this is basically just getting points. But of course, like for this, you would actually like need the funding organizations to actually play ball and properly recognize that. Yeah, and since there aren't, there are many people doing antibody research, yeah, but generally like I don't think that all that many to actually get like this kind of like global recognition. But yeah, generally just raising awareness of the funding bodies like would be my guess.
I guess the answer to your question would be that you would like to have some kind of metric on societal impacts or the level of collaboration, or bringing something to a next level rather than restricting it to your own situation and building it on metrics on the individual researcher. But I think that's also the difficulty, because what type of way should we do that unless the EU or other funding bodies start to recognize this as an important thing? And that's what we need to bring up to them then, probably. But I guess looking at how things have gone, that it's simply easiest to take the simplest metrics and work with that. And I think it takes a whole lot of talking and discussion to try and change that system.
So one possible solution for that, maybe that, I don't know, besides the citations and acknowledgements in the publications and also in the funding applications, will be one part in this publications, the data usage, besides the data availability, maybe data usage. If we have the, for example, the number for each data set, the unique number like the one for the publication, that we can, in the data usage part, we can clearly indicate what data we are using. That can be linked to original data sets. And then this can be easily calculated from the metrics. And metrics can be used for the other, using like funding applications, something like that. That could be used for the recognition of the data set owner or data set generator.
Okay, so which would directly mean that we need the data itself to become citable, like with a DOI. So which then would mean that databases in general should not only have a publication saying, okay, the database is out there, but that each individual data set is there so that it's attractive for people to have well annotated data sets so that they are used by other people, and so that this data set is then also used.
Yeah, so I want to add that this is already being in realistic. So I don't know whether you know a journal which is called Giga Science. That's the one mostly published genomic data. That's also this code generated or the established by BGI. So that journal, just like what I mentioned, I have every data set citable, have a unique identifier for each data set. So that's the scope of that journal, is to make a data set as much reusable as possible.
Yeah, I would comment some of these things you're talking about. These are sort of challenges that I think are being faced globally by a lot of different high throughput data types, right? So you have this in the microbiome community. But I think one thing to keep in mind is that there are other communities that have sort of successfully created environments where it is the expectation that data is shared, and journals put a requirement that those data types are shared. So I do think that as a community, we should be able to get there within the current environment. I mean, just look at the transcriptional profiling world. I mean, you can't publish, you know, a microarray or an RNA-seq study without making your data publicly available. Just the reviewers would never allow it, the journals would never allow it, and I think we should be able to get to the same place as a community here.
No, I completely agree. It's the question on how we do the change management, because I mean, probably the people that are here are the first 20% who are willing to share, and so the question is, how do we get the next slice of people on board? And those are the people who still need incentives within the classical academic system. But I think making data sets citable and making sure that they are citable and that people get direct credit for it would be probably one of the most simple and direct ways for people to understand how what they get out of it.
Question on the topic of metadata. We've talked a lot about annotations. I'm wondering if there are stories that people have of using data that somebody else generated, especially data that's generated by somebody you don't know. What's the risk tolerance of people using someone else's data to do research?
Well, we have done a study where we combined data that we have generated ourselves and then combined them with data that others have published to online repositories. And I will talk about that tomorrow a little bit. But you have to be careful and you have to spend a lot of time exploring that data and to see what difference do you have, where do they come from, and how to adjust for it. So, I mean, that is something you have to spend quite some time before you can actually do analysis, but it does work. You know, if you're careful, that does work.
I mean, that was kind of the goal of minimal standards to some degree, to facilitate that. You're exactly right. You have to, if you have the data there in front of you, you can start making that judgment.
So let me just bring up too that through the Antibody Society, there's a guy named Daniel Emmerling who is starting an initiative to create a survey that I think would go out to everybody in tabs and probably beyond. So that's 1,500 members, 60% of who's in industry, asking people about do they have data sets, and if they do, are they willing to share them, and if not, why not? And so they'd still have to put that survey together and probably test it on some focus groups. But if anybody's interested in helping Daniel with that, let me know. And I think I've pointed him to the AIR community as a place to go to help him put that together. So just thought I'd bring that up.
I think we have 10 more minutes on the schedule at least.
Okay, then probably one final thing, and switching gears a little bit. So we now talked a lot about public data, but initially there was also talk about private data, and of course how that is attractive to a commercial in a commercial setting. So looking at the pharmaceutical industry, I mean, they, of course, usually their small molecular libraries are one of their most secret treasures and assets that they have. Nevertheless, they found ways to compare those libraries in kind of zero knowledge fashion against each other, so to find out whether they're working on the same compounds. Is this something that you would envisage would make so this kind of the capability of databases to allow these things would be something that would be attractive or make us more attractive to industrial partners?
I mean, if I can start off on that, basically, if we got data like that then it would be great. Yeah, because generally, like I know from working with many pharma companies that like they are basically just sitting on just gold mine of data. Yeah, incentivizing them to actually release it is really tough. Yeah, because as you said, this is something that like is their property. So even after commercialization, they really just prefer to hush about that. And secondly, for them, it's basically just work to just release it. But basically, just having said that, like there are people in many companies that are actually just really willing to just participate in some data sharing activities. Yeah. So for instance, we were involved in something called Advance project, which was basically just it wasn't NGS. It wasn't AIRR-seq data. It was basically just releasing structures of antibodies within pharmaceutical companies, and some of those people basically just release their structures, and mostly like their incentive was that there was a promised publication at the end of that. Yeah, so basically like people in companies sometimes like they're also incentivized by the fact that like if they get a publication that they basically just can get a bonus at the end of the year. So. But generally, like with releasing their phage display libraries and so on, I don't see much chance, to be honest.
So, regarding the sharing of the private data, so besides the concern from the from the pharmaceutical companies, there are also concerns from the individuals. So I think there's still an open scientific question about whether the AIRR-seq data can be used as the molecular identifier for each for individual identities. So if this is the case, that will be, like what we deal with the genomic data. And some other, so I think that this scientific question have to be solved. That will be useful for talking about sharing private data.
If I can just say, basically, I do remember some papers that actually like were saying literally that you can distinguish individuals just by looking at their repertoires. So yeah, like in the moment that like we can actually just link it back to an actual named individual, this is like a really slippery ground.
So that may be true, but that same issue exists with RNA-seq data, and yet there's still wide sharing of RNA-seq data. We just have different repositories with different levels of protection in order to deal with that.
And it works for genomes as well. I mean, so, and you imagine an immune repertoire would be less identifiable than a genome because it changes constantly. So your immune repertoire in a year is going to be different than your one currently. So your genome should be roughly the same. And so you'd think there'd be a way we can do it.
I think because this is something that in terms also of data privacy, of course, if we treat every AIRR-seq repertoire as complete genomic data, and we try to get broad consent from the donors, then we're good. The question is there a possibility also to share data where we do not have that? And I think this is something that also as a community we should discuss, because in the end it comes down to a very simple question: how much entropy is in that thing? And if we're talking about more than 30 bits, then we simply have to assume that we can re-identify people on that planet. So then clearly we're going across this. But I think we as a community are probably the people that are best suited to actually make that to make that judgment at some point. And this is probably also something worthwhile working on, because at some point policymakers will ask us, okay, is this something that we need to protect? And I think we have the tools. We can estimate the diversity of the loci and thereby the potential re-identifiability. And the other question, of course, is that besides who you are in the genome, the question is also, what have you done? I mean, if there is an antibody signature, whether you had syphilis or not, well, it's not that sure whether everyone wants to have that out there in the wild.
Thank you. So it's there's going to be a suggestion to have a new working group that would be basically legal and ethics here in the community, and some people who are willing to lead it are Jake Shirkow and Tanya Bubella. We've got to wait till Tanya gets here to talk about it a little bit more. But again, if you have any interest in that type of discussion or being part of that sort of group, look up me, and I can point you to Tanya when she gets here. Yeah.
So now it's time to wrap it up, I guess?
Yeah, so, but there is a last question from Yuri.
I don't think we have time.
Until his 10 questions ticket expires.
I mean, I was going to say, are we not like all just lying to ourselves to a large extent? Where, you know, I've written some IRBs, and I've basically been told that if you're doing RNA-seq, as long as you're not doing straight up genome sequencing, it's not identifiable, like quote unquote. And but it seems pretty clear to me that it's very easy to identify. I mean, people actually use RNA-seq data. They mix cells in single cell sequencing, and then you know, unmix them just by looking at the genotypes. And I suspect that the same thing will be true exactly with immune-seq data, except it'll be even worse, because now you will know if someone has syphilis if we're successful as a group in you know, being able to phenotype people based on your immune repertoire. So, and I mean, it seems obvious that it's extremely identifiable and much worse than even something like RNA-seq. On the other hand, you know, if people are not going to go to jail for posting a bunch of, you know, RNA-seq data, I suppose we're just a subset of RNA-seq. So maybe that's okay.
Yeah, but I guess the question is very simple. I mean, if we think that the data is sensitive, then we put it in the ICF, okay? And with that one, no one is going to jail, and our institutions won't be sued for 4% of their yearly income, according to GDPR. So I think then it would be fine. But then at least we've done the due diligence, and I think that's what it is about, to have an answer that is really scientifically sound, and not as you just described, where we say, well, we're claiming that it's not identifiable, but everyone who does the calculation on the back of the envelope sees, okay, well, this is clearly identifiable because you're covering 30,000 SNPs.
Is it too late to add just a subset of RNA-seq to the AIR logo? Is that?
So before we thank this great panel, I wanted to mention we're going to power through 2 more reports from our communications subcommittee and then our meetings subcommittee. Then we get to move to the other room and get a reception and the tool demonstration. So we'll have all the things you're looking forward to, but we have these 2 more reports, and then let's thank these guys. Great.